Good morning, students. So today in this video, we will be discussing the checkpoint. So last day we have discussed that how the checkpoint facilitate in rectification of errors in DNA, such that the proper DNA goes to the mitotic in the mitosis process, and there is no malicious effect of those DNA. So today we will be discussing a checkpoint on your early G1. Now there is one, I have talked about tumor suppressor. Tumor suppressor are those which basically suppress the tumor. If you remember the cancer genetics, I have talked about that there are two types of mutation. One is the gain of functional mutation and another is the loss of functional mutation. So conversion of proto-oncogene to oncogene is a gain of functional mutation. But if I talk of your loss of functional mutation, loss of functional mutations are the inactivation of P53, inactivation of tumor suppressor, which will initiate the process of cancer. So RB. So let's move on to the molecular biology of it. That is how they regulate this particular cell cycle checkpoint. So RB was first identified as a product of a gene responsible for retinoblastoma, which is a rare childhood eye tumor. So it is an eye tumor, right? Further, it was also found that if you inactivate RB, if you mutate RB, it will not only produce retinoblastoma, but it is also associated with several different types of cancer. Now the question is, how this retinoblastoma protein is associated with the cell cycle checkpoint. Now, oncogenic protein like RAS and cyclin D derive cell proliferation, and the protein encoded by many tumor suppressor gene include RB. So you see what happens. This RB is basically interacting with E2F. E2F is the elongation factor 2, which is basically being used in the transcription process, helps in the elongation process in transcription. So when RB interacts with your E2F, so when this particular complex is being formed, what is happening? E2F is not being able to respond properly. E2F is not being able to work properly. E2F is inhibited. So transcription is repressed. But in presence of CDK4, 6 and cyclin D, this particular thing, in the previous class I have told you that whatever is happening in your cell cycle is the two tools are very important. That is your CDK4, 6 and CDKs and cyclins, cyclin dependent kinase and cyclins. So what will happen? This RB is phosphorylated. So once RB is phosphorylated, RB dissociates from E2F, and once E2F is free, E2F is active. So when this E2F is active, E2F is actually activating the transcription. So again I repeat that RB is responsible for retinoblastoma. Mutation of RB not only results in childhood tumor but also associated with different types of cancer. Now this RB is associated with E2F. So RB in its active form will bind with E2F, and thus the E2F will remain inactive. E2F is the elongation factor 2, which will remain inactive, and as a result the transcription will be repressed. But when in presence of CDK4, 6 and cyclin D, the RB will be phosphorylated, and as RB is phosphorylated, this E2F opens up, E2F is relieved, E2F is free. So E2F now gets active and the transcription factor activation begins. Now the question is, what will happen when this transcription will begin? Now, that progression through the restriction point and entry into the S phase. So it's basically the restriction point between G1 and S. So when they move on to the S, this is mediated by CDK2 and cyclin E. So here the participating factor is your CDK2 and cyclin E. So what is happening? I told you that this particular transcription which we are talking about. So what is happening to this transcription? What transcription is happening? So this transcription which is happening is the synthesis of your cyclin E. Cyclin E is stimulated by E2F followed by the phosphorylation of your RB. Now, activity of CDK2 and cyclin E. So CDK2 and cyclin E forms a complex, but they are inhibited in G0 and G1 phase by an inhibitor, that is your P27. And this P27 belongs to CYP-KIP family. So this is also another family which is inhibiting the activity of CDK2 and cyclin E. So increased synthesis of cyclin D leads to binding of this P27 to CDK4/6 cyclin D. So P27 will now bind with CDK4/6 and cyclin D complex, relieving CDK2 and cyclin E. So once CDK2 and cyclin E complex relieved, they are activated. So they will activate some helicase, which is MCM helicase, which will unwind the DNA at the replication origin and will start the DNA synthesis, and thus the cell cycle will progress from your G1 to S phase. So let's see the diagram. From the diagram you get to see that CDK2 cyclin E is bound with P27. So P27 is the inhibitor. So P27 is the inhibitor, so it has inhibited the whole process. Now, on the other hand, you know that this E2F is actually being activated by phosphorylation of RB. So that will increase the cyclin E synthesis, and more cyclin E will bind with CDK2, but they cannot activate because of P27. So you have to repress down the P27. So how to repress down the P27? You have to increase the synthesis of cyclin D. So once cyclin D is synthesized, P27 has more affinity towards cyclin D than cyclin E. So cyclin D binds a complex CDK4 and 6. So this whole complex will be formed. That is cyclin D, CDK4, CDK6, P27. And once this complex has been formed, in late G1 you get to see more and more of CDK2 and cyclin E complex, which is an active complex. And once this active complex is being formed, they will now activate this MCM helicase. So MCM helicases are basically activated, and this will unwind the replication origin, resulting in formation of your replication bubble, starting the replication, moving from your G1 to your S phase. So I repeat once again. So here we have started from E2F. So E2F is actually activating transcription. So transcription of what? Transcription of basically cyclin E. So once the cyclin E is formed, cyclin E forms complex with CDK2. Now CDK2 cyclin E complex is an active complex which will allow the progression of the cell from G1 to S. But there is another inhibitor which is your P27. So P27 will inactivate it in the form of a complex. So first you have to repress down the P27. So for repression of P27, you have to increase the synthesis of your cyclin D. So once cyclin D is synthesized, now the P27 will have more affinity to cyclin D rather than cyclin E. So cyclin D, CDK4, 6 and P27 forms a complex, whereas CDK2 and cyclin E is relieved. So in late G1, you will have more and more of your CDK2 and cyclin E. And that will enhance your MCM helicase, activate your MCM helicase, which will unwind the DNA at the replication origin, resulting in the movement of the DNA from your G1, but not movement of the DNA, movement of the cell from G1 to S phase. So this is your G1 to S phase transition. Next, we will move on to the other damages which can be caused by the DNA. So you know, you get to see double-stranded break and your single-stranded break. Now double-stranded break and single-stranded break both will activate this ATM and ATR. These are also kinase. Okay, so ATM and ATR are the kinase which will phosphorylate this particular protein CHK1 and CHK2. So ATM will phosphorylate CHK2, whereas ATR will phosphorylate CHK1. Now once this two is phosphorylated, they will inhibit. You get to see this blockage sign. This is inhibition. Inhibit CDC25 from getting activated. Why? Because CDC25 in its activated form will activate CDK2 and CDK1, and they will result in progression of the cell from G1 to S and G2 to M. So if CDC25 is stopped, the activity is stopped, then it will inhibit. CDC25 is deactivated, so CDC25 will deactivate or inhibit the activation of CDK2 and CDK1. And so as this CDK2 and CDK1 activation is stopped, there is a G1 to S arrest by CDK2 and G2 arrest by CDK1. So this is how your double-stranded break and your single-stranded break bring about your G1S arrest and G2 arrest. So double-stranded break activates ATM. ATM will phosphorylate CHK2. This will block CDC25, which is a phosphatase that will basically activate your CDK2 and CDK1. But if you block it, then they will inhibit CDK2 and CDK1 and resulting in G1S arrest and G2 arrest. Next, I come to the last point, that is your P53. We call it P53 as a guardian of the genome. P53 is a tumor suppressor gene. In our next classes, we will also see what are the different roles which are played by P53. So till now, let's discuss this, that P53 activates your ATM, again kinase, which will activate CHK2. This will phosphorylate P53. So P53, which was once inactive, will now get activated, and more P53 will get phosphorylated. So this P53, once phosphorylated, they will activate P21. This is another tumor suppressor. So P53 indirectly activates P21. And then P21 will bind with CDK2 and cyclin E, inhibiting CDK2 and cyclin E, forming that inactive complex which we have discussed previously, resulting in G1 arrest. So again I repeat, P53 is a tumor suppressor gene. So P53, once there is a break or there is a damage of DNA, P53 is activated, ATM is activated, and that will activate CHK2, which will further phosphorylate your P53. So more and more P53 will get activated, activating your P21. And as this P21 is activated, this P21 now bind with CDK2 and cyclin E together to form a complex, and that will bring about your G1 arrest. So that's all for the class today. Tomorrow we will be discussing, that is the last class, we will be discussing the spindle assembly checkpoint, and that will end up your cell cycle checkpoints.